Welcome to Medical Lab Science Quick Review. We will continue our series about basic concepts of autoantibodies that cause autoimmune diseases that appear in our ASCP study guide. Two very interesting autoimmune diseases that we will introduce to you are called autoimmune hepatitis and primary biliary cholangitis. The liver is the largest internal organ and gland in the human body, resting in the right upper quadrant of the abdomen, inside below our diaphragm to the right of the stomach, and shielded by the right lower rib cage. The gallbladder, also known as a cholecyst, is a small pear-shaped bladder tucked below the liver that stores and secretes bile produced by the liver. This diagram shows the anatomic relationships of the liver, gallbladder, biliary tree, pancreas, and its pancreatic duct to the stomach and duodenum. Here we see only a partial list of the numerous major liver functions. It has been estimated there are about 500 functions of the liver, also known as our body's chemical factory. For your information only, cholecystokinin is the major hormone responsible for gallbladder contraction and secretion when bile is needed for fat digestion in the duodenum. For a specific topic, we'll only focus on these anatomic parts. Autoimmune hepatitis was formerly known as leukoid hepatitis or autoimmune chronic active hepatitis. In our ACP study guide, a question refers to chronic active hepatitis associated with a specific autoantibody that you are required to know. It is interesting to note that other etiologies besides autoimmunity may cause chronic active hepatitis. FYI only, let's discuss liver histology with a pathologist. Hematoxylin and eosin stain demonstrates hepatocytes organized into plates, lobules, sinusoids, portal triads. Phagocytic immune Kupffer cells may be visualized. While the pathologist examines the pathology slides, we'll focus on the serological testing. Note numerous autoantibodies associated with autoimmune hepatitis. According to the question in the study guide, focus on one autoantibody, which is the anti-smooth muscle antibody. What is smooth muscle? Smooth muscle myocytes are spindle-shaped with wide middle parts and tapering ends with a single nucleus. They are shorter than striated skeletal myocytes. Inside the sarcolemma membrane, there are muscle proteins, actin and myosin filaments attached to dense bodies, all of which mediate contraction and relaxation, similar to that seen in skeletal muscle cells. The important distinction of smooth muscle cells are that they are innervated by ganglion of the autonomic nervous system. We don't have to think about their contraction and relaxation. Anti-smooth muscle antibodies attack smooth muscle tissues throughout certain parts of the body. They are seen in some cases of chronic active hepatitis, some rheumatic disorders, and some infectious diseases. When anti-smooth muscle antibodies attack the liver, this leads to autoimmune hepatitis. Immunofluorescent patterns in anti-nuclear antibody testing or ANA testing demonstrates a speckled pattern. As we can see, a speckled pattern in ANA testing might be helpful but not specific or diagnostic. Primary biliary cholangitis was previously known as primary biliary cirrhosis, and the autoantibody is anti-mitochondrial autoantibody. Anti-mitochondrial antibodies attacks intrahepatic bile ducts, leading to chronic cholestasis, inflammation, ultimately to liver cirrhosis. Now we are ready for two questions and answers.